And good afternoon, everyone. Welcome to Research Grand Rounds. I'm Dr. Tim Allen Williams, Chair of Medicine here at University of Louisville. And it's my pleasure to introduce our speaker. I know it's Thursday, and many people will be looking at this online, and if not live. But we're going to have a wonderful discussion about emergency granulopoiesis as a regulator of neutrophil effector function. And it will be given to us by Dr. Marcin Wysoczynski, who is an associate professor of medicine here at the Center for Cardiometabolic Science, and actually within our division of environmental medicine in the Department of Medicine here at University of Louisville. Our background, we're so pleased to have you here and to hear from you. His background is actually from Krakow, Poland. And he worked his way through in Poland for his master's and Ph.D. He's done his postdoctoral fellowship here at University of Louisville, and he's worked his way up from instructor of medicine, assistant professor, and now associate professor with tenure. So congratulations on all you've accomplished so far, and we're looking forward to all you have to teach us.
Thank you, Dr. Williams, for kind introduction, and I'll go through our recent discoveries on the role of emerging granulopoiesis as a regulator of neutrophil function. So, as a member of Center for Cardiometabolic Science, my research is centered around the cardiovascular disease, and my research lab is focusing on understanding the role of immune system in pathophysiology of heart failure. Today, we're going to be talking about neutrophils, and in general, immune systems. So it's been well understood that we evolve in environment which is a pathogen rich, and we develop mechanisms to protect our body from those pathogens, and that was very fairly hostile environment with a lot of pressure to develop mechanisms which will protect us from external elements and pathogens. And we have three basic levels of how our body is protected from the pathogens, which is first, it's just a very simple barrier, external or internal barrier. This is skin, epithelium, and some of the basic mechanisms which will protect us from invading organisms. And second level of protection is once the primary immunological barriers are compromised and pathogens invade our tissues, we have activation with so-called innate immune system. And that immune system is composed of cells like in the case of macrophages, basophils, neutrophils, and monocytes. And those cells are produced in large amount daily. And they don't require any instructions or training to perform their function. They possess pathogen recognition receptors, which can recognize stress, pathogens, or damage to the tissues, and they can deploy their effector functions. We have also adaptive immune system, which requires a little bit more training. Those are B cells and T cells, which secrete antibodies, are also specifically recognized to specific antigens, and they're being instructed by innate immune cells to perform the effector function. They provide long-term protection from organs once they're fully trained. So today I'm going to be talking a little bit about innate arm of the immune system. And for many, many years, it's been thought that immune system evolved just to protect us from invading pathogens. As I mentioned, when primary immunological barriers are compromised and pathogens are invading our tissues, they release PAMs. So those are pathogen-associated molecular patterns, and that could be recognized by immune cells, but also secondary to the invasion of immune cells and pathogens, it could be observed damage to the tissue, which deploys proteins from damaged tissues and damaged cells called DAMPs, and they're called danger-associated molecular patterns. So you can recognize that the immune system primarily evolved to protect us from those organs, and more recently has been recognized that the immune system plays an important role in sterile inflammation, tissue repair, and regeneration. Although the observation that immune cells infiltrate injured tissues without pathogens was observed over 100 years ago by clinical pathologists, but you observe the immune cells reside in the tissues like myocardial infarcted hearts. However, initial thought was that immune cells appear in those tissues by accident and they just cause damage. And in 70s and 80s, there have been a lot of studies trying to study what is the extent to which immune cells cause damage, and interventions to limit inflammatory response with using broad immunosuppressants were failed to produce any substantial results in protection of the tissues after myocardial infarction, especially, or stroke. And about 15, 20 years ago, it has been recognized that actually immune cells which are recruited to infarcted tissues after myocardial infarction, they are beneficial and they're responsible for healing of the myocardium. And it has been also recognized that imbalance in this immune response could have detrimental effects. So although it is well-orchestrated response, when that response is over-exacerbated, it could cause tissue damage, but a lack of that response is also detrimental for healing. So on the forefront of that response is neutrophil. Neutrophils are the most abundant cells in human blood. They're produced in large amounts, and they are fast responders that appear in the sites of inflammation, regardless of the cause, if those are bacteria, viruses, or ischemic injury to the tissues. They appear as the first responders. So one of the common characteristics of infection or steroid inflammation due to ischemia is neutrophilia. All the neutrophils are very abundant in peripheral blood of healthy individuals. Their number could drastically increase in response to infection or tissue injury. And also one of the association of that immune response is to increase diversity of neutrophil phenotypes. Some of the phenotypes which are normally not present in humans, they appear in acute inflammatory response. And there are a few sources of neutrophilia reasons. So first, it's just a persistence. So in response to PAMPs and pro-inflammatory cytokines, the neutrophil lifespan is extended. So their disposal is delayed and they can just persist longer. And that is one of the reasons why we have more neutrophils. We have also demargination of neutrophils from the pole, which is deposited in the vascular beds of the lungs. There's a large population of tissue neutrophils in the lungs, but also in the bone marrow. Neutrophils in the latest stages of maturation, they reside in the bone marrow, and in response to stress, they're being deployed to peripheral blood, but also granulopoiesis. So it's been demonstrated that in response to cytokine storm, there is increased rate of neutrophil production, and those are main contributors to neutrophilia after MI. So what is granulopoiesis? So granulopoiesis is the production of neutrophils in the bone marrow. Primarily, it occurs in the bone marrow, where hematopoietic stem cells differentiate to granulocytes, monocytes, progenitor cells, and they further are being advanced through their differentiation to band cells and maturing neutrophils. And upon maturation, those neutrophils are being released to peripheral blood for immunosurveillance, and they don't reside in peripheral blood too long. In humans, it's been calculated that neutrophil resides in the circulation for less than 1 day. And they're constantly infiltrating, marginating into tissues to perform immunosurveillance. The recent studies by Andres Hidalgo showed that that occurs in the circadian manner. So neutrophils are being deployed fairly early in the morning, and several hours later, they're being deposited to virtually all tissues, and they probe those tissues for potential damage or infection. So upon infection or tissue injury, neutrophils deploy their effector function, and it is important to understand how those functions are being deployed, because that is a constant balance between their effector function, which could be bacterial killing or clearance of the infarcted tissue or damaged tissues, and that's a balance between also tissue damage. So their effector function, which are positive, could be balanced and outnumbered by negative impact and tissue damage. So understanding how those effector functions are regulated could be beneficial for the future therapeutics. And in general, terms, we can divide those effector functions into two, intracellular and extracellular. So intracellular mechanisms are related to phagocytosis. So neutrophils can phagocytose bacteria, but they can also phagocytose smaller particles due to tissue damage and necrotic bodies. And phagocytose particles are being killed. So if those bacteria, they're being killed by reactive oxygen species, which are being released to phagolysosomes, but also they release proteolytic enzymes and antibacterial peptides. And those neutrophils, which with embedded bacteria, is being picked up in silent matter by macrophages. But in the abundance of tissue damage or tissue inflammation, neutrophils can deploy extracellular mechanisms of killing, and this is oxidative burst, and neutrophil produce abundance of reactive oxygen species, and they can degranulate the primary, secondary, tertiary granule content to extracellular space, which is enriched in antibacterial proteins, but also proteases, which are important not only in bacterial killing, but also tissue remodeling. And as a last resort, neutrophils release what's called neutrophil extracellular traps. So that is a situation when neutrophils cannot handle bacterial load or amount of necrotic tissue. They activate the program, the NETotic program, and what causes that is together with ROS and PAD4, histone become modified, citrulinated, and being cleaved by proteases that cause decondensation of chromatin and release to extracellular space. So those nets are decorated with histones, which are highly immunogenic, but also decorated by antibacterial proteins. And this is the way neutrophils can trap bacteria and curtail further expansion of the infection. So what is important to mention, that the majority of those functionalities are being programmed during granulopoiesis. So at the end stages of neutrophil maturation, neutrophils are fully equipped with those functional mechanisms. So they're being fully equipped with all the NADPH oxidase components, mitosis components, also degranulation, all the granule content being deposited in neutrophils during the maturation state. So they are responding to tissue damage or inflammation within minutes. So they don't require transcriptional reprogramming like macrophages to acquire their phenotype. All the programming is happening during granulopoiesis. So in the situation of infection, majority of neutrophils extravasate to infected or damaged tissue, and those neutrophils deploy their function. That creates the vacuum because the marginated neutrophils from the bone marrow are released in the peripheral blood, and that what is causing to activation what is called emergency granulopoiesis. So emergency granulopoiesis, by definition, is increased production of neutrophils in the bone marrow, and that is happening by increased proliferation of neutrophil progenitors and precursors and faster maturation. So that causes increased production of neutrophils, which are released in peripheral blood, and they provide the second wave of neutrophils, which are infiltrating tissue and also causes sustained neutrophilia. So the current view of emerging granulopoiesis until recently was that emerging granulopoiesis just provide in response to cytokine storm like GCSF, GM-CSF, IL-3, IL-6, or those granulopoietic cytokines can increase production of neutrophils just to increase their number. And so today was not really recognized, what's the role of emerging granulopoiesis in diversification of the function. So we asked question, to what extent does emerging granulopoiesis modulate the neutrophil effector function? So we developed the assay in our lab to ramp up of neutrophil production and increase emerging granulopoiesis by injecting GCSF. So in those mice, we injected 100 micrograms per kilogram per day of GCSF for 3 consecutive days to increase their production in the bone marrow. So after 3 days, majority of the neutrophils which were produced by a normal granulopoiesis were replaced by neutrophils which are produced by emergency granulopoiesis. So in the first step to validate that, we wanted to validate we actually observe neutrophilia. And what we observe that 6, 12, and 12 hours post last dose of GCSF injection, we saw observed neutrophilia, which was the first good sign that our model works. And one of the characteristics in which bone marrow respond to cytokine storm is to release of immature neutrophils, which is called left shift. And we quantify those immature neutrophils in peripheral blood by flow cytometry and using CD11 negativity. So mature neutrophils express CD11, immature don't. And we found that we have a release of immature neutrophils from the bone marrow in response to GCSF. So that's a good sign that injection of GCSF, which mimics cytokine storm, can cause neutrophilia, but also bone marrow response to release some of those immature neutrophils. So next we wanted to quantify if we actually have activation of granulopoiesis. So we injected mice with EDU, and EDU will incorporate in proliferating cells during granulopoiesis. So we can quantify a number of neutrophils which incorporate the EDU, and that will tell us if we have actually increase of the production. So what we found that in response to GCSF, we had about 50% increase in neutrophil production in the bone marrow. So we validated the model that 3 doses of GCSF injection causes activation of neutrophilia and activation of emerging granulopoiesis, and that's going to cause accumulation of neutrophils in the bone marrow, which are produced by emerging granulopoiesis. So next we wanted to assess their function. So one of the first functionalities which is deployed by neutrophils is ROS production. This is mainly fueled by NADPH oxidase. We could measure the magnitude of the ROS production by dihydrorhodamine. So rhodamine is changing upon stimulation of ROS. It's fluorescing. So we can measure the fluorescence by flow cytometry. And in our model, we use PMA, which is a PKC agonist. So neutrophil responds to PAMs and DAMs and dangers and from extracellular space by binding the receptors and signaling. All the signals are found through PKC. So this is like a hub to deploy their neutrophil effector functions. So we use PMA, and we observe that in response to PMA, neutrophils produced by normal granulopoiesis produce abundance of ROS, but those neutrophils produced by emerging granulopoiesis have impaired ROS production. So that could be very interesting observation, which could lead that second wave of neutrophils coming in could have reduced ROS production, could have potentially impaired bacterial killing mechanisms, but also can limit tissue damage. So one of the second assays we deployed to quantify neutrophil function was to measure netosis. So we use 2 DNA binding compounds. One is Hoxt, which is cell permeable. So pretty much Hoxt goes to the cells and to label nuclei of every single neutrophil. So we could quantify total number of neutrophils in the view. And we use Cytox green, which is also DNA binding compound, which fluoresces when binds extracellular DNA. So what we found that in response to calcium ionophore, which is a very robust activator of netosis, we see increased netosis, which you can see in green. So this is vehicle injected mice, and those are normal neutrophils produced by normal granulopoiesis, and they robustly respond to calcium ionophore to produce nets. However, neutrophils produced by emerging granulopoiesis have an impaired net production. So we have 2 components of neutrophil function which are being reduced now under ROS production and netosis. And next neutrophil function is deployed during the site of inflammation is degranulation. Neutrophils have 3 types of granules: primary, secondary, and tertiary. And those neutrophil granules contain antibacterial peptides, but also proteases and other compounds which are important in signaling to macrophages. So what we did, we stimulated neutrophils with PMA, and we collected conditioned media to measure primary granule content. We found that neutrophil elastase, which is a primary component of primary neutrophil granules, it's secreted in more robust manner in response to PMA. We also measured some secondary, tertiary granule component content. They release them in normal manner, so there's no preference in secretion of secondary and tertiary granule content. However, primary granule content was exacerbated in neutrophil produced by Marcin Wysoczynski. And finally, neutrophils can also produce cytokines, which orchestrates monocytes recruitment and macrophage polarization. And we measure three chief pro-inflammatory cytokines and chemokines, which is TNF-alpha, IL-1, and CCL2. So CCL2 is a chemokine, which binds CCR2 receptor on monocytes and is very robust chemoattractant to monocytes, which come to the site of inflammation after neutrophils. And what we found that those neutrophils produced by Marcin Wysoczynski, they have a heightened production of TNF-alpha, IL-1 beta, and CCL2 in response to toll-like receptor agonists, which is LPS. So what we found so far that we have decrease of ROS production and ptosis, but we have increased secretory function, which potentially could have enhanced immunomodulatory properties. So we wanted to test, that communication between neutrophils and monocytes and macrophages in a peritonitis model. So what we use our GCSF model. So we had two cohorts of mice. One was treated with vehicle, so they can have neutrophils which are produced by normal granulopoiesis. And we treated mice with GCSF for 3 consecutive days to replace those neutrophils with emerging granulopoiesis neutrophils. And subsequently, we inject those mice with zymosin. Zymosin is a particle which is derived from yeast wall. So it's primary toll-like receptor 2 agonist, which can elicit inflammatory response in the peritoneum. So here in blue, as just demonstrated immune response in normal mice in response to zymosin, what we observed that we have increased total leukocytes with robust infiltration of neutrophils and monocytes, and when we stratify those monocytes to Ly6C high and low monocytes, and subsequently those monocytes differentiate to macrophages. So in this particular matter, we think that those Ly6C high monocytes, they infiltrate and differentiate to macrophages. An intermittent state of macrophage population is those Ly6C high monocytes. So that's going to predict our trajectory of monocytes infiltrating tissue and their commitment to macrophage lineage. So in response to zymosin, when mice which were treated with GCSF, we observed a little bit more robust response of neutrophils to zymosin, but what we also observed that we have a little bit faster trajectory of those monocytes differentiating to macrophages, and as a consequence, at 48 hours, we also observed twice more macrophages in tissues compared to vehicle-injected mice. So this classical view that emerging granulopoiesis in response to cytokine storm is increasing neutrophilia, and now we can be a little bit revised. And now what we observe that those neutrophils produce in response to cytokine storm, they're a little bit different. And what we found, those neutrophils produce less ROS nets, and they're more susceptible to degranulation of primary granule content, and they respond to toll-like receptor agonists to produce more cytokines, which orchestrate monocytes and macrophage response. So in the next, we ask, what is the particular mechanism? What causes that reprogramming of neutrophil function, those fairly fast responding elements to the tissue inflammation and injury? And so we perform RNA sequencing. So we collected neutrophils from normal mice and mice injected with GCSF. So we can have two groups of neutrophils from normal granulopoiesis and emerging granulopoiesis, and we subject them then to RNA sequencing. We found that they transcriptionally they're fairly different. So that is represented here by PCA plot analysis. So this is vehicle group. Those neutrophils are quite different than neutrophils which were isolated from mice injected with GCSF. So when we did individual gene expression profile, we found that over 3,000 genes were differentially regulated. And when we grouped them, there was a lot of genes which were upregulated, a lot of genes which were downregulated. Majority of the genes which were altered in neutrophils produced by emerging granulopoiesis were related to cell metabolism. And majority of those genes were related to glucose metabolism, glycolysis, and we decided to engage in collaboration with Dr. Hill, who is our metabolism expert, to further explore what would be the difference in neutrophils produced by emerging granulopoiesis. What's the difference in metabolism? and how this metabolism changes could drive their effector function. Neutrophils primarily utilize glucose as a carbon source to produce energy, and that has been demonstrated, over 5 decades ago in very simple experiments when neutrophils in vitro cultures were exposed to glucose and other carbon sources with fuel other metabolic pathways, and only glucose was able to support bacteria killing neutrophils in vitro. So it has been demonstrated that glucose is a primary source of carbons to produce ATP and produce energy. Now, and also neutrophils, which are produced by normal granulopoiesis or normal neutrophils, were exposed to full growth media with all the carbon sources, and inhibitors of mitochondrial complex 1 complex 3 did not alter ATP levels. So that further suggests that mitochondria is not involved in ATP production, and inhibition of oxidative phosphorylation also showed no difference in ATP production in those neutrophils. So second pathway which was discovered to be involved in neutrophil functions is pentose phosphate pathway, and that is branched from G6P. And main function of this pathway is to regenerate NADPH, and NADPH is a fuel to NADPH oxidase to produce reactive dominant oxygen species. And ROS is one of the main component of bacteria killing mechanism when we inhibit PPP pathway, and this was also published by others. Inhibition of PPP pathway, it will increase number of surviving bacteria in the cultures of neutrophils with bacteria. So that's a readout showing that PPP pathway is a very important component of bacteria killing mechanisms. So mitochondria has been demonstrated to not be involved in ATP production, and however, mitochondria play a very important role in regulation of intrinsic apoptotic pathways. So mitochondria contain a lot of pro-apoptotic proteins, which are contained within mitochondria when the mitochondrial membrane potential is maintained. And when mitochondrial membrane potential is lost, those proteins are being released and neutrophils undergo apoptosis. And more recently, it's been recognized that mitochondrial membrane potential can be also maintained by G3P shuttle, glycerol-3 phosphate shuttle. So when electrons can be directly fed to complex 3 of electron transport chain mitochondria, is going to maintain mitochondrial membrane potential and potentially increase survival of neutrophils in response to stimuli. So now we were lucky enough to have metabolism experts in our group, Dr. Hill and Dr. Lorkiewicz. Dr. Lorkiewicz is our expert in mass spectrometry. He was able to perform metabolomics in neutrophils. So this is just to understand how in our hands neutrophils culture isolated normal mice, how is their metabolism, and what are the accessory pathways of glucose metabolism which are involved in regulation of neutrophil functions. So we use C13 labeled glucose, so that's universal labeled glucose, so all carbons are labeled C13. And after stimulation, we're able to extract those metabolites at different time points, and we can track the glucose incorporation to each metabolic pathway. So what we found, we confirmed that we have 2 time points, 10 and 60 minutes. So they respond very fast to stimulation, to PMA. And we confirmed that majority of carbons go through PPP cycle, and they go very quickly through. So that confirms observation from literature that PPP cycle is very active, very involved in regulation of neutrophil function, and especially those killing mechanisms by NADPH oxidase ROS production. We also confirmed that we have carbons going through the glycolysis pathway to culminate with the lactate production. So also we found, which that's a fairly new pathway, which is involved in the regulation of neutrophil function, is glycogen. A lot of those carbons, which are derived from glucose, are being incorporated into glycogen. As you can see, we have almost 100% of enrichment of this glucose label into glycogen, and that happens very fast. And more recently, it has been recognized that glycogen is a store of extra, fuel, neutrophils, and can be deployed really fast. So in environments of low glucose, like during infection or ischemic injury to the heart, this glycogen provides the boost of energy and fuel for neutrophils to perform the function. And we also found that in response to PMA, we have increase in some of the TCA cycle metabolites. So that indicates that all the neutrophils are not very abundant, but they do have a certain level of mitochondrial metabolism, probably not involved in ATP production, but maybe it's involved in preservation of mitochondrial membrane potential and regulating the apoptosis. And we also observed that this new pathway, glycerol phosphate shuttle, is actually fairly active, feeding electrons to complex 3. And if you can appreciate the difference between proportion of carbons into glycerol phosphate shuttle compared to TCA cycle, it seems like glycerol phosphate shuttle is more active in neutrophils produced by animal granulopoiesis compared to TCA cycle metabolites. So in next step, we wanted to see what is the temporal change in abundance of those metabolites. So what we found that at 10 minutes, we have a fairly early increased abundance of pentose phosphate pathway metabolites and glycolysis metabolites, and also glycerol phosphate shuttle metabolites. And on the other hand, TCL cycle metabolites were down, so it means they're being utilized and they're being metabolized to downstream metabolites, but the carbons are not flowing through those pathways enough to restore their total abundance over seconds. So they're being utilized, but not being replaced. And at 60 minutes, we observed that some of those PPP cycle metabolites being further metabolized to downstream pathways, and we have decrease in abundance of also glycolytic pathway, although they're still upregulated. And we have further decline in metabolizing TCA cycles. It seems like glycolysis is very active, but there is temporal change in those pathways because they flow through those pathways and metabolites are flowing farther down but they're not being fastly replaced. So it seems like there is some temporal regulation and shift in where the carbons go and how they are being replaced during neutrophil activation. So we recently published this paper about phase partitioning of neutrophil oxidative burst, and we wanted to see we wanted to investigate the role of accessory glucose metabolism pathways in the kinetics of oxidative burst. So we use XF seahorse to measure rate of oxygen consumption. So that's going to be a readout for oxidative burst. And this is just an example here when neutrophils are stimulated with PMA. This is time kinetics of oxygen consumption. We have a very fast oxidative burst, which peaks and slowly resolve, but neutrophils still continue utilizing oxygen, and we suspect that it produce reactive oxygen species. So that gives us opportunity to study what is the temporal change in oxidative burst in response to when we intervene with individual pathways. So the first pathway which we intervene was the glycogen storage. So we use glycogen phosphatase inhibitor. So that's an enzyme which will mobilize glycogen and liberate glucose to feed the PPP cycle. And what we observe that when we inhibit glycogen mobilization, oxidative burst it's a little bit delays, declines. So it's not as really robust as it is when glycogen storage is available. So next we block complex 1, mitochondrial complex 1. We observe there's no difference in oxidative burst in those neutrophils, so that further support the fact that TCA cycle and maybe complex 1 is not involved in regulation of oxidative burst. However, when we block complex 3, we observe that there is a prolongation of oxidative burst, which suggests that complex 3 is potentially involved somehow in resolution phase of the oxidative burst. And when we use glycerol 3-phosphate shuttle inhibitor, we observed that entire response of neutrophils to PMA in oxidative burst was declined. So glycerol 3-phosphate shuttle seems like it's the chief regulator of that response in neutrophils. So we developed this functional model. So that's in normal neutrophils. And now that allows us to do a couple of things. Now, once we understand what is the glucose metabolism in normal neutrophils, we can study what are the changes which drives neutrophil function in glucose metabolism in EG neutrophils. So we found that glycogen is involved in the initial burst of ROS production, and that is further supported by glucose metabolism, glucose uptake and metabolism, and mitochondrial complex 3 plays some kind of role in resolution of that response. However, glycerol 3-phosphate shuttle is the master regulator of that response, and that gives us the opportunity to maybe modulate glycerol 3-phosphate shuttle activity to temper normal neutrophil function and reduce their reactive oxygen species production and perhaps mitosis. So now we perform one experiment to see what is the difference in mitochondrial content in those neutrophils produced by emerging granulopoiesis, and we treated mice with GCSF sorted neutrophils. We compared the mitochondrial content with neutrophils produced by normal granulopoiesis, and we found that they have a higher abundance of mitochondria, which is shown here by higher expression of complex 3 and 4 in those neutrophils, but also higher citrate synthase activity, which is actually it's going to be relating to mitochondrial mass in those neutrophils. So those neutrophils produced by emerging granulopoiesis, they have a higher mitochondrial content. We still don't quite understand what would that mean, how that translate to neutrophil function, but we developed this functional, working hypothesis that carbons flowing through glucose acetyltransferase pathway feeds glycerol 3-phosphate shuttle, and that feeds complex 3, and some of the carbons can go through TCA cycle. And what we found that neutrophil supply during homeostatic conditions rely on G3P shuttle to maintain mitochondrial metabolism, and due to higher abundance of neutrophils produced by emerging granulopoiesis, we think that EG neutrophil supply neutrophils that shift with increased TCL cycle flux to maintain mitochondrial metabolism. And that's going to be the main component which is going to drive the difference in their functional outcomes in regulating bacteria killing and tissue repair after tissue injury. And now we're just doing experiments actually test this hypothesis and figure out physiological consequences. So today we can revise further that hypothetic model that EG neutrophils in response to cytokine storm undergo a metabolic reprogramming, and they contain more mitochondria and have more active TCA cycle, less active glycerol 3-phosphate shuttle, which results in less ROS production, less net production, but increase and sustained degranulation and cytokine production, and they orchestrate in a more effective way monocytes recruitment and macrophage polarization. So next we wanted to test what is actually the physiological consequence of that reprogramming, and we evolved in this pathogen-rich environments, and there was strong pressure to develop those immune responses, including emerging granulopoiesis, which those neutrophils can orchestrate monocytes recruitment and macrophage polarization and perpetuate the inflammatory response, which is necessary for bacterial killing. They have to be very effective in killing of the pathogens invading. But now the question is, now our lifestyle has changed a little bit. Now we live in sanitary conditions. We are protected from this pathogen to a certain degree. But in return, we develop more modern diseases like arteriosclerosis and myocardial infarction leading to heart failure. And now the question was, how does initially favorable evolutionary pro-inflammatory actions translate to microarray repair and progression of heart failure? So inflammation plays a very important role in myocardial repair. So in normal inflammatory response leads to infiltration of neutrophils, which deploy their functionalities. They orchestrate monocytes and macrophage responses, which remove necrotic tissue and remodel infarcted myocardium, and subsequently they recruit fibroblasts, which proliferate, produce collagen, and the results we have a scar formation, which preserve myocardium from rupture and dysfunction. So as a result, we have this durable scar formation, which decline ventricular function, but it's necessary to preserve the further dilation and progression of heart failure. So if that, and it has been demonstrated that any disturbances in this immune response, so if this response is over-exacerbated, the quality of the scar formation is impaired, and the scar is thinner and ventricles larger, and that leads to acute heart failure. And in the same situation occurs when this immune response is insufficient to remove necrotic tissue. It's just healing response is delayed. So we decided to study the role of emerging granulopoiesis in post-myocardial infarction wound healing. So first, we subjected mice to MI. We want to confirm that we see neutrophilia was going to be the first characteristic of the systemic response. So mice subjected to MI, we collected blood from them, and we measured neutrophils, and you can see 24 up to 4 days after MI, we have increase in neutrophil count, which is a characteristic of neutrophilia, and that neutrophilia is sustained up to day 4. And we have also release of immature neutrophils from the bone marrow, so that suggests that we have a systemic response, including bone marrow niches responding to myocardial infarction. And second, we want to confirm that we have activation of emerging granulopoiesis. So the mice subjected to MI were also injected with EDU, and we measure the rate of neutrophil production. We found that mice subjected to MI produce about 50% more neutrophils than those mice, naive mice or some sham subjected mice. So we have activation of emerging granulopoiesis. So now we have a model, so we can start emerging granulopoiesis is activated in our MI model, and we wanted to find some genetic interventions so we could block that emerging granulopoiesis response, so we could test what is the effect on post-MI wound healing. So normal granulopoiesis is orchestrated by transcriptional factor CBP alpha. So CBP alpha is expressed on common myeloid progenitor cells, and that directs those common myeloid progenitor cells into neutrophils, and it's important in equipping neutrophils in their functionalities. And in the absence of CBP alpha, mice are neutropenic. They don't produce neutrophils. However, those mice still respond to infection or tissue injury. So although they're neutropenic, injection of E. coli staph or some other components which drives inflammation will increase number of neutrophils in peripheral blood. So it means there's some compensatory mechanism in stress which will overcome neutropenia due to CBP alpha deficiency. It has been demonstrated that this factor is CBP beta. So in mice which in the absence of CBP alpha, mice subjected to stress or infection, the CBP beta is upregulated and can take over of granulopoiesis. However, in CBP beta knockout mice, CBP beta and the mice subjected to infection, they don't produce sustained neutrophilia. So it has been demonstrated that this cytokine storm driven by GCSF, GM-CSF, or cytokines signals through JAK-STAT to upregulate CBP beta, and that is the main gatekeeper of emerging granulopoiesis. So we cross mice, so we have a CBP beta plus mice, and we cross them with S100AA transgenic mice to have neutrophil-specific CBP beta knockout. So this particular crease expressed fairly early in granulopoiesis on the level of promyelocytes. So that's beneficial in our model. And we confirmed that knockout. And we also wanted to see if we're going to have impaired neutrophilia in response to stress. So we use GCSF or cytokine to induce emerging granulopoiesis, and we observed neutrophilia was reduced. This study is underpowered. That's a very recent study. but also we have a decrease in release of immature neutrophils in peripheral blood. So there is a high probability that those mice will have impaired emerging granulopoiesis. So we subjected those mice to MI. We want to see, our initial expectation was that emerging granulopoiesis will provide a supply of neutrophils, which potentially pro-inflammatory actions, they recruit monocytes and macrophages, they dysregulate the immune response. However, in our surprise, we observed something opposite. Male mice subjected to MI, in the absence of emerging granulopoiesis, they just die. Out of 15 mice, 10 died in male group, and majority of those deaths were due to LV rupture. So they don't have proper infarct healing mechanism in the absence of neutrophils produced by emerging granulopoiesis. Although females survive, and we had the same survival in females, we didn't see any difference. When we performed echocardiographic analysis, we found that the stroke volume, cardiac output, and ejection fraction was lower. So those mice survive, but they do much worse than mice which have normal emerging granulopoiesis. So we've revised our model, and now some of this data was generated in different models of emerging granulopoiesis. Now we want to have a functional outcomes. So in response to cytokine storm, CBP beta deploys emerging granulopoiesis responses, which reprograms, which cause metabolic and functional reprogramming neutrophils. And that reprogramming is important for myocardial repair after MI, and this is more pronounced in males than in females. So we have a few minutes. I want to discuss a little bit another facet of our studies is that how actually bone marrow sends the tissue damage in the heart. So this is our hypothetical model that after myocardial infarction, dams are being systematically released, and we see neutrophilia. And we still don't understand quite how neutrophils or progenitor cells in the bone marrow is a sense of injury in the heart. So one of the important characteristics of hematological profile in the peripheral blood in response to MI, but also to different acute inflammatory responses, temporal lymphopenia. So it has been demonstrated in several studies that in patients and in mice that initial burst of neutrophilia is accompanied by decline of circulating lymphocytes in peripheral blood. And that has been something described in other forms of viral and bacterial infections, that the temporal lymphopenia occurs. So it could be explained by a few different mechanisms how that happens. So first is that neutrophils just inhibit. So we inhibit lymphocytes production. So we have bone marrow granulopoiesis is enhanced, and that declines lymphocytes production. Second mechanism could be just in response to glucocorticoids, endogenous glucocorticoids. Some of the lymphocytes undergo apoptosis, and that's why they decline in their number, or they're just being deposited in the tissues, and that could include whatever tissue is inflamed or could be deposited in other peripheral tissues. And we stumbled upon a couple of papers showing that actually T cells are very important regulators of hematopoiesis in the bone marrow. So we hypothesized that perhaps that lymphocytes from peripheral blood could home to the bone marrow to orchestrate those emerging granulopoiesis responses. So we subjected mice to MI, and we profiled immune cells in the bone marrow. We found that acute after MI, within 24 hours, we had a slight decline in myeloid cells, which could be potentially explained that they're being mobilized, and granulopoiesis did not catch up yet to increase their production. But we have increase in lymphocytes in the femur. And when we stratified those lymphocytes to B and T cells, we found that only significant population increase of T cells in the femur of the mice subjected to MI. So that could be due to not only homing, but also the production in the bone marrow. To further test that, if this is related to their homing, what we did, we isolated lymphocytes from spleens, and we labeled them with green dye FSC, and we injected them into peripheral blood at the time of myocardial infarction. And 24 hours later, we quantify green cells into the bone marrow of those mice. As you can appreciate here, the number of those green lymphocytes increased sixfold in the bone marrow, and we stratified them further to subsets of T cells. We found increase in CD4, CD8, also gamma delta T cells in the bone marrow of the mice subjected to MI. So now we are not really trying to explain lymphopenia, but we're trying to explain what's the mechanism of communication. So that is the first step to understand T cells, which are responding to myocardial infarction, and they're potentially being exposed to some of the circulating dams homing to the bone marrow. Next, we want to test if they're going to be actually responsible for neutrophilia. So what we did, we depleted lymphocytes prior MI. So prior MI, mice were injected with anti-CD3 antibody, which depletes T cells. The mice were subjected to MI, and we achieved near 100% depletion of T cells in the peripheral blood. But also what we observed that at 1 day and 3 days post-MI, we had completely abolished neutrophilia in those mice. So that suggests that those circulating T cells, they respond to some stress in the heart and some cues in peripheral blood, they're being redirected to the bone marrow, and they instigate emerging granulopoiesis to produce neutrophils, which are beneficial for post-MI wound healing, the preserve function. So that's all I have for today, and I would like to acknowledge my lab. So majority of this work was done by 2 of my students, John Stefan Joe, and my lab manager, Taylor Zabul. But also, I would like to thank the director, Dr. Jones, who is an integrative physiologist and expert in all the models of mouse models of heart failure. I would like to thank Dr. Hill, who was the major driver of the metabolic studies in those neutrophils, and Pavel Lorkiewicz, who is expert in proteomics. And I would like to thank Physiology Core for myocardial infarction models and functional analysis of the heart, and Richard Singal from our bioinformatics course. And I would like to thank also our funding mechanism, which make that work possible. Thank you.
So thank you so much, Dr. Wysoczynski. This is going back a number of years in my career because I was thoroughly annoyed by white cells. Going into myocardial infarction, my research was fluorine 18-deoxyglucose for hypertonic myocardium. Going into the coronary care unit with a single head detector, which we had souped up the physics to actually measure fluorine 18-deoxyglucose. It is acted on by hexokinase that is phosphorylated, and then that's it. And so it becomes a permanent ion marker for viability of a cell if you can get it to take up. And so we're looking for, what area of the penumbra zone is still alive and is going to recover if we can bring back blood flow. And instead, we're getting white cells, which are taking up the FDG. But now, after your presentation, I realized maybe we should be going back to that because the prognosis of the patient is going to be not just the recovery of the penumbra zone, but the strength of the scar. And they're both going to have the FDG. So anyway, just to say that out loud, we are going to have a PET center here. We will have access to FDG, typically for inflammation and infection in the myocardium, but we probably should go back to infarction. That's like 40 years ago for me. But
yeah, that's actually the field is revived a little bit, especially the neutrophil field, when initially thought there was just neutrophils are homogeneous. And now it seems like the neutrophils are not homogeneous. There are different flavors, like monocytes and macrophages, and they could play diverse roles in that wound healing response. So I would be also interested in collaborating with some physicians, especially in STEMI patients, to look for the subsets and measure the function in temporal resolution of their mind. And if we could have a follow-up, 5-10 years later, to see how these patients do in relationship to the neutrophil functionalities, that'd be very important too. And studies, I think that'll be very interesting, especially from my perspective.
Oh, that'd be fantastic.
And if we can also correlate that with some neutrophil counts and functions in the myocardium, that would be even more robust and very interesting.
That'd be great. We have some questions online, or anyone else?
Nothing yet.
Okay. So I guess Yeah, there's probably people are thinking of inflammation, infection, and getting out of 30.
Yeah.
But this was really stimulating for me, just because the white cells are so important. Getting rid of the junk so that you can actually form a good scar. And I don't think we recognize how important that is without the kind of work that you're doing. So
there's a lot of facets we are also interested in working in the future. So one of the question is, which you guys being easy on me, because one of the relationship between the neutrophilia and outcomes after MI, it's those patients which have higher neutrophil counts are doing worse. So this is a little bit counterintuitive. So emerging neutrophils is necessary. But this model could be more complicated. So we have more flavors of those neutrophils. There's not just switch from one to another phenotype. That's number one. And I'm interested to actually look at the aging. So majority of our work was done in the young mice. And it seems like in young mice, this is very important. But actually, majority of the aging population, they don't handle infections very well.
Right.
And there's a relationship between patients which are aging patients and subjected to MI. Older they are, they do worse. And a recent stroke study show that although emerging neuropathy responses are occurring in response to stroke, but they result in production of dysfunctional neutrophils. There is something disconnected with aging, how the body responds to infection or injury. So I'll be interested also looking at the aging population relationship of their neutrophil function and outcomes of their MI.
One of the functions of this conference is to stimulate further research, and we have an interested resident here. What you just said, I think we should have a Wysoczynski index, which is the white blood cell count divided by the troponin level. Because what you're saying is that people with a higher white count have a poorer prognosis. That's probably because they're having a larger myocardial infarction, which we're actually measuring with the area under the troponin curve. We do that routinely. Suppose you had, a low troponin and a very high white count, then you're probably going to get more repair. And if you have a low white count and a high troponin, you're in trouble.
Yeah, or maybe when different subpopulations of those neutrophils will be different. So we can identify the phenotypes which are responsible for activation of emerging granulopoiesis in relation with function.
Right.
If we can develop that in patient population, that would be beneficial for the future understanding of the initial diagnosis of are those patients will be doing okay, or there'll be some additional interventions could be deployed to help them out. So I think that's my future interest. Probably I'm not going to resolve all the problems next.
Right. Yeah. Well, the FDG thing was ruined because we started revascularizing everybody. So we don't care how much viability there is. We're going to give you a shot at it by opening the artery, and that's the way it should be. But I bet that there's some necrotic tissue, and that we should be able to measure those two parameters very simply because we're measuring them in everyone. Let's correlate them with outcomes. But
I'd also be curious if there would be a threshold of physiologic aging fibrosis of the bone marrow where you see this emergency granulopoiesis decrease. I don't know if there's, and that's looking more at what we think about. But because our patients tend to be older, their bone marrow is not functioning the same way. We would see it in a young mouse. And I think the aging component would be really important because that's who we see.
Exactly,
and who's having MIs and who's having larger MIs.
Yeah, and also I'm interested in effects of exercise. So if we could also have a correlation between muscle mass, skeletal muscle mass, and outcomes, because it seems like more muscle is better in humans. They're healthy, but also immunosenescence and muscle mass is going right to head. So there's future directions and potentially can have
absolutely
conversation to explore.
I think that's very insightful. That whatever index you could come up with for infarct size versus WBC response in the peripheral blood, you're going to have to measure something, some parameter such as ejection fraction or left ventricular size. We could actually measure scar tissue, particularly with MRI. You can measure how much actual scar there is. All of those parameters are going to be dependent on a lot of things like aging and exercise. But we should be able to dissect that out.
Yeah, absolutely.
Yeah.
So it'd be really interesting.
Quick comment from online here from Dr. Emmons in BMT. He says, oops, scrolled up on me. I'll get it in a second. It says, He says the WBC is tricky. Keep in mind that most neutrophils are in the tissues with low circulating pools. Hence, it's tissue neutrophils that really matter. He says, not sure what the surrogate marker for that would be. And then he goes on to say the effect of the GCSF contributes to overall immune suppression in mixed T cell assays. He said it may also alter mobilization into the tissues with rising levels in the blood. Hence, the blood levels per se may be misleading.
Yeah, so that's the known crosstalk that GCSF. So we have increased activation of neutrophils, and they're known to be T cell suppressive somehow. But that does not exclude the possibility that T cells homing to the bone marrow. We don't know still what is the population, what's the preference for the T cells going to the bone marrow. We think this is some low level of glucocorticoids, which redirect them from infarcted tissues to from peripheral to the bone marrow. We still testing that, and we don't know if they're going to be activated T cells. We think they're activated T cells, but we still don't know what's the population and how they're going to be orchestrating neutrophilia. We still don't know that, but those studies are ongoing. It's a good comment.
That's all.
All right. Well, thank you so much.
Thank you.